Welcome to the deep dive. Today, we're headed to Las Vegas, but uh, not for the usual reasons.
Not for the casinos.
No casinos this time.
Eh?
We're diving into something a little less glamorous, the city's sewer system.
Oh, okay.
Yeah. There's this fascinating study about what we can learn from wastewater.
Interesting. Sounds a little messy, though.
Right. Definitely not a typical vacation spot, but there's gold in that, wastewater data.
I can see that.
One of our listeners actually sent us this research paper. It's about pathogens and these indicator trends they found in Southern Nevada wastewater.
Mm.
It covers the time before, during, and after the COVID-19 pandemic.
Oh, wow. So they're looking at how the pandemic maybe affected things.
Exactly. And they want to know, like, what are the key takeaways from this? What did they actually find?
Makes sense. Wastewater can, tell us so much about the health of a population, like almost like getting a secret look into the city's health diary.
Totally. That's a great way to put it. This study, it was done by the Southern Nevada Water Authority.
Okay.
And they looked at wastewater from 6 major treatment plants.
Wow. That's a lot.
Yeah, it covers over 2 million people.
So it's not just a small sample size. This is like a significant chunk of the population.
It is. They tested for a whole bunch of stuff. I mean, like viruses, bacteria, you name it. They were looking for adenovirus, enterovirus, norovirus, Cryptosporidium, Giardia.
Oh, wow. The usual suspects.
Right. And then some things I'd never even heard of, honestly.
Yeah.
Yeah. Things like pepper mild model virus
Pepper mild model virus.
and crassphage. Ever heard of that?
Crassphage. Yeah, actually, that's a pretty interesting one. I mean, it's a virus, but it infects bacteria.
What?
Yeah, specifically the bacteria that are found in, the human gut.
Okay.
So it's become this really helpful tool for tracking fecal contamination.
Oh, so like kind of like a breadcrumb trail.
Exactly. But a microscopic breadcrumb trail, it can lead researchers back to the source,
Right. Back to the source.
And the cool thing is it's really specific to humans. So it helps researchers tell the difference between, is this contamination from humans or is it from animals?
No, so it can distinguish.
Yeah.
Yeah.
Like if they find crassphage in a water sample, that's a pretty good sign that there's been some human fecal contamination there.
Okay. I get it. That's actually pretty smart. So they're using this virus to track other stuff.
Exactly. Very clever, isn't it?
Yeah. Very clever.
Yeah.
So in this study, they used 2 main ways to analyze the wastewater.
Okay.
One is qPCR,
Right.
and the other is cell culture.
Uh-huh.
Can you break those down for us? Like, what are those exactly, and why would they use both?
Sure. Okay. So imagine you're a detective, right? Looking for evidence.
Okay.
qPCR is like using a really, really powerful magnifying glass.
Okay.
You're searching for like tiny traces of a virus's genetic material.
So like DNA and stuff.
Exactly. DNA or RNA. And it's super sensitive, so it can detect even the smallest fragments.
Oh wow. So it can tell you the virus is there even if there's just a teeny bit.
Exactly. That's qPCR.
Right. Okay. And then cell culture.
Cell culture, that's more like growing a plant from a seed.
Okay.
You take a sample of the wastewater
Right.
and you try to actually grow live viruses
Oh.
in a controlled environment, of course.
Right.
And this tells you not only that the virus is present, but it tells you how much of it is actually infectious.
Oh. Okay, so that's the difference.
Yeah. One tells you it's there, one tells you if it can actually make you sick.
Okay, that makes sense.
Mm-hmm.
So they're getting like 2 different pieces of the puzzle
Exactly.
from the same sample.
Hmm.
qPCR is like, okay, how widespread is this thing? And then cell culture is, okay, but how much of a threat is it?
Precisely. You've got it.
Great. And then they didn't stop there. I mean, these researchers, they were really thorough. They wanted to make sure their data was super reliable.
Oh, good science. Always a good thing.
Right. So they went back and retested some older samples.
Oh, wow.
Yeah, they had stored them just to be sure their results were consistent.
Like a double check.
Yeah, like a double check to make sure everything was accurate.
Smart. I mean, especially if you're going to use this data for, like regulatory decisions or things like that, you want to be extra sure.
Absolutely. So, okay, enough about the methods. Let's get to the good stuff.
Results.
Yeah, the results. What did they find? Anything surprising?
Oh, definitely some interesting stuff. For example, they found higher concentrations of norovirus GI than norovirus GI.
Isn't GII usually the more common one?
Yeah, usually. In most clinical settings, you see more GII.
Right.
But in this study, GI was like the star. Yeah. So that raises some questions. Could GI be causing more infections than we realize? Maybe those cases are just not being reported as much.
Huh. Like a sneaky virus.
Kind of. Flying under the radar.
Interesting. And what about this whole thing with super shedders? That caught my eye.
Super shedders, yeah, that's a pretty memorable finding.
It is. So what is a super shedder exactly?
Well, it's not a superhero, that's for sure.
Right.
But these are individuals, they shed a lot more of the virus than the average person.
Oh, so like one person can really skew the results.
Yeah. They found that, particularly in the smaller facilities, these super shedder events, they could cause these huge spikes in the concentration of viruses.
Wow. So one person can have a big impact.
They can. It really shows you how individual behavior can influence the bigger picture, especially in those smaller communities.
Okay, so sneaky viruses, super shedders.
Right.
What else did they find in this wastewater? Any other surprises?
Well, remember during COVID,
Yeah.
all the focus on hygiene, all
Yeah.
that hand washing and social distancing,
Right.
did it actually make a difference? Like for other viruses,
Like they looked at that.
they did. They compared the levels of a few viruses,
Okay.
adenovirus, enterovirus,
Uh-huh.
and both types of norovirus.
Okay.
They compared the levels before the pandemic and during the pandemic.
Got it.
And what they found is actually pretty amazing.
I'm going to guess.
Go ahead.
Levels went down during the pandemic.
Exactly. Way down, sometimes like by a factor of 100
Wow.
or even more. It really shows you,
Yeah.
those public health measures, even though they were, a big change,
Right.
they had a real measurable impact.
Yeah.
They actually reduced the spread of these pathogens.
So it actually worked, the things they told us to do.
It did. Sometimes common sense and science, they actually line up.
Sometimes. That's good to hear. So this study, they didn't just focus on Las Vegas, right?
Right.
They also compared their results with a big project in California.
Right.
It was this Water Research Foundation project.
Yeah.
Project 4989, I think.
That's the one.
They were looking at pathogens across the whole state.
Right.
I'm curious, did they find the same kinds of trends?
There were some similarities,
Okay.
but also some interesting differences.
Oh,
For example, they noticed that overall the pathogen concentrations were higher in southern Nevada.
higher than California.
Yeah.
Wow. That's kind of surprising.
It is interesting, but we have to be careful about, jumping to conclusions.
Right.
Remember, the Southern Nevada study, it covered a longer period of time.
Okay.
And that included, before the pandemic, during the pandemic, and after the pandemic.
Right.
So that could explain some of the variation.
Yeah.
Plus, different areas, they have different populations, different diets, even different plumbing systems.
Right.
All of that stuff can affect what ends up in the wastewater.
Okay, so it's not a perfect comparison.
Not exactly. Apples and oranges, maybe?
Right. What else did they learn about, like how these pathogen levels change? Did they find any patterns?
They did. Remember those qPCR tests?
Yeah.
The ones that can detect even the tiniest little bits of viral genetic material?
Right. Super sensitive.
Yeah.
Yeah.
Well, they noticed some interesting seasonal variations.
Seasonal, so like more viruses in the winter.
You got it. That's a common pattern, especially for viruses like norovirus.
Right.
They tend to spread more easily in the colder months
Okay.
when people are, spending more time inside.
Right. Huddled together,
Yeah.
sharing their germs.
Exactly. They found that both GI and GII, those two main types of norovirus, they were significantly higher during the winter.
Okay. That makes sense.
Yeah. But they also found some seasonal variation for adenovirus and enterovirus, although not as dramatic as norovirus.
Got it.
Adenovirus was slightly higher in the winter,
Okay.
and enterovirus, it seemed to peak in the fall.
Interesting. So it's not just norovirus we got to watch out for.
Right. It's like you can't let your guard down when it comes to hygiene, but especially in those winter months.
Yeah. Wash your hands, people.
Yeah.
What about those fecal indicators they were tracking? Did they find anything interesting there?
Yes. Remember crassphage?
The reliable one?
That's the one. The virus that helps us track human waste.
Right.
They found it in high concentrations in all the samples.
Okay.
So, confirming it's a pretty reliable fecal indicator.
Good.
PMOV, that's another commonly used indicator.
Right.
That one was also consistently detected.
Okay.
But HF183, that's a bacterial indicator.
Okay.
That one showed some unexpected fluctuations.
Fluctuations, like what does that mean?
Well, in October 2022, the levels of HF183, they suddenly dropped across all the treatment plants. It's a bit of a mystery. The researchers are, they're still trying to figure out what caused it.
Yeah.
They think it might have something to do with maybe with how they were measuring it.
Oh. So maybe it's like an error.
Could be, or maybe it's something else entirely. They're not sure.
Interesting. So even in science, sometimes you hit a snag.
You do. It just shows, how important it is to be transparent about the limitations of a study.
Right.
And to always question the findings.
Always question. So, okay, we've talked a lot about the details of this research, the nitty-gritty.
Yeah.
But let's zoom out a little bit.
Right.
Think bigger picture. What does it all mean? What are the implications of all this? Like, why should we care about what's in Las Vegas wastewater?
Good question. Well, for one thing, it has big implications for water management, especially in areas that are, facing water scarcity.
Right, like the desert Southwest.
Exactly. This kind of data, it gives regulators really valuable information for setting water reuse standards.
Like for drinking water.
Especially for direct potable reuse.
DPR.
Yeah, DPR, where they take wastewater and they purify it to drinking water standards.
Oh, wow. So this research could actually help make sure our water supply is safe.
It could, especially, in those drought-prone regions.
Yeah, okay, that makes sense. So we've got implications for water.
Right.
What else?
Well, it also has huge potential for public health surveillance.
Oh, right.
Imagine, being able to monitor wastewater for pathogens and detect outbreaks early on. Even before people start showing symptoms.
Oh, wow. So like an early warning system.
Exactly. It could revolutionize how we respond to public health threats.
That's amazing. Okay, so we've got water management, disease surveillance, anything else?
Yeah.
What are some of the bigger questions that this study raises?
Well, this study, it mostly focused on the liquid part of wastewater.
Okay.
But there's another component that might be important.
Another part?
Yeah, the solid fraction.
The solid part. Wait, isn't wastewater all just, liquid?
No, actually, there's quite a bit of solid material in wastewater.
Oh.
And that solid fraction, it likely contains a lot of those pathogens. And those fecal indicators.
So they're missing a piece of the puzzle.
They might be. That's actually one of the points they raise in the study.
Okay.
They suggest that that future research should focus on measuring pathogens in both parts.
Both the liquid and the solid.
Exactly, to get a more complete picture.
Okay. Yeah, so it's like we're only seeing half of it right now.
Kinda. We've got this whole other dimension to explore.
Wow. That's pretty cool. I mean, it's a little gross, but it's cool.
Right. And the insights we could get from studying both fractions, I mean, it could be incredibly valuable for public health, for source tracking, even for understanding how well different wastewater treatment processes work.
Yeah. That's a great point.
Yeah.
So much potential. It's amazing, right? Like all this information, all this data, just hiding in something we usually flush and forget about.